We're all suited up here,
Yeah.
and we're about to enter what exactly?
Okay, so you're about to get a tour of the world's largest autonomous lab.
Okay.
We call it Nebula.
Nebula.
And it's 52 of our rack automation carts, so 52 robotic arms, connected to about 70 pieces of laboratory equipment.
Okay. RAC is an acronym, RAC, is that right? What does that stand for?
Reconfigurable automation cart.
Reconfigurable automation cart.
Yeah.
Okay, there we go. All right, let's check it out.
Yeah, let's do it.
All right, Jason, thanks for having us down here today at your lab space in Boston.
Yeah, thanks for coming. It's gonna be super fun.
For sure. So when I think about and listen to the founders of the large frontier AI labs, whether it's Dario Amodei or Demis Hassabis or so forth, when they talk about, what the net benefit is of these billions of dollars that are being invested into AI, invariably they go to science. And often it's life science in particular. And for example, Demis on a 60 Minutes interview last year said, gosh, within the decade, potentially, we're going to solve all disease. Very exciting. Also, it's one thing to build intelligent systems, but it's another thing to actually have those systems meet reality and meet biology. And so one reason I'm really excited to be chatting with you is that you have a lab space that's building very large autonomous biology systems that actually do the science. And when I think about organizations that have leaned maybe the hardest into making the bet on the future, I think of Ginkgo and I think of your work as the CEO. So excited to explore and learn more about what you're building and the path that you took to get here.
Yeah, super excited. Yeah, and I do think it's a unique time. We just announced the project with OpenAI maybe about a month ago where GPT-5 ran that lab. So I think it's a great time to be talking.
Okay, well, I definitely wanted to get into that. Maybe before I do, the audience might notice there's a beautiful, looks like a T-Rex face here. So yeah, let's start off with this. What are we looking at?
Okay, this is my most prized possession, by the way, just so you know. So when we started, I'm in biotechnology because of Jurassic Park. Let's just be clear. I was 13 years old when the movie came out. I was a Michael Crichton book nerd, right? And so I was obsessed. And I went to MIT for undergrad, and I was like, I want to be a genetic engineer. And there was no bioengineering major, so I actually did biology and chemical engineering because I figured chemistry was close to biology. It took me 2 years to realize it's oil and gas plants. But it was fine, no problem. I'm in the lab, I'm trying to do this stuff. And so it was very much what brought me into this, right? And we had shirts made when we started Ginkgo that said, had the Jurassic Park logo and said Ginkgo Bioworks. And then on the back, it said, there will be dragons. That was our punchline, all right? So we take the company public back in 2021. And the folks we did the listing with, they were Hollywood folks, at least one of the guys was. And he's like, I got a present for you. It's the day of the IPO. It was already a pretty good day in my life. If you see, come take a look, Nabil. So here's the dinosaur head, but what's this say? It says, Jason, you are right. There will be dragons. Steven Spielberg.
Look at that.
I mean, come on, right? And so I literally the greatest thing, the greatest gift I've ever received. So anyway, that's what that is, yeah. But I do think if you, I'm 45, right? And so my age biotechnologists, shocking number, are doing it because of Jurassic Park. So I think this thing, I have an 11-year-old now, and I think the things we're exposed to at that time, like fiction and this consumer sort of focused things that help inspire folks, it makes a big difference. It makes a really big difference. I think people don't really realize that, but it certainly does. And so anyway, so that's why we're here.
Cool. Well, thank you for sharing that story. And a good just reminder of the importance culturally of not just science communication, but storytelling in science.
100%. It is underestimated. Yeah. And I think you're seeing this now with all the AI stuff, and we can talk about the stories around how it'll be applied to science. But how we communicate technology and how we communicate science with the public, I think Jurassic Park is actually one of the best stories of science because it merges both majesty and the technology. You're like, I want dinosaurs. Right. But at the same time, humility in the face of powerful tech, right? Life finds a way. And Crichton was just perfect with that, right? And so that to me is the ideal. That's kind of how you want to communicate these things, right? Not just doom and gloom, not just a Terminator or something, but this sense of there's majesty in this, but also if you don't approach it with humility, you blow up.
Well, let's talk about some of you mentioned OpenAI, and you've had a few, let's say, concrete examples recently that I think help bring this to life. The first, I'll start off maybe the oldest of the three that I have in mind, which is with PNNL. So you had a anaerobic microbial phenotyping platform,
Absolutely.
AMP2,
AMP2.
which experimentally perturb and assay biological systems in an anaerobic environment. So tell us more about what that means and what the purpose was of that build-out.
So I had the real honor to, in December, do a ribbon cutting on this new system. It has 18 of our robots. We're going to get to go show them to you later in the lab for the Department of Energy and this lab, Pacific Northwest National Lab. And what that lab does, the PNNL, is it was the lab that was focused on like cleaning up atomic waste. All right, so it's sort of like the cleanup national lab. And so the biologists in that lab do a lot of work with microbes that live in the soil. And so this particular lab is designed to work with anaerobic microbes, in other words, microbes that can't live in air. They need no air. And so what you can imagine, that's a hard environment for humans to work in. And so the way that it's done today traditionally is you have like an anaerobic chamber, which is like a box that you could pump all the air out of. You put your hands in gloves and you do all your work like in a very difficult environment to try to do it with these gloves. What a great place for robotics. And so I think one of the interesting things that project shows is a great pull to get started with particularly like, we'll talk about in a minute, like autonomous labs that are much more complicated than traditional automation is in environments that are not great for humans to work in. I think anaerobic is a good one. So that's how that started. But Secretary Wright was there because it was part of the Genesis Mission, which is this new effort out of the White House to bring AI into science and in particular into the national labs. And that's done both through partnerships with the frontier labs, put all the data, from NASA into the model and see what it can learn. But it's also being done with companies building laboratory robotics because part of the idea is we're going to need to make new data and models need a lot of data and that's going to need automation. And so we were there at opening the autonomous lab. And then we actually also announced 97 more robots they were buying in a $47 million contract. That'll happen over the next couple of years.
Wow. Congratulations.
Yeah, it was really awesome. Yeah, it's cool. We can talk a lot more about it, but yeah, it was neat.
So you mentioned data, and one of the things that caught my eye is that one of the benefits of the system is that it helps generate, it says genotype to phenotype data sets.
Yep.
And so, often when I think about, I visited the Broad sort of lab or visitor center recently, and they talk about how different parts of our DNA can then code for different diseases, and if we can understand that link between genes and disease, that's very beneficial for humanity. Yep. We're talking about anaerobic, microbial life, but what link is there? Well, one, can you tell us more about that research? Actually, maybe we'll start there.
Yeah, and I'll step back because I think like when these AI companies are talking about like bringing AI into science, it's worth asking just sort of like, how do you do science? Like, what does that even mean, right? And so there's certain parts of science like mathematics, theoretical physics, simulation, where everything is running, essentially can be run inside of a computer, right? And you are trying to discover a new, Terence Tao is trying to come up with some new math theorem, and then it will be tested computationally. And at no point do you need to go really interact with the real world. It's all theory. That's a very small fraction of science. So the overwhelming majority of science is experimental because you can't theorize your way to understanding how that anaerobic microbe does this thing or that new material does this thing or whatever. You can have some theories that inform you, but eventually you hit a limit of what you can understand based on what is currently known about the world and what has been done by the theorists. And at that point, you enter, even in physics, the world of experiments. Yeah. Experimental physics, right? Particle accelerators. Well, biology is dominantly an experimental discipline. The way we learn how biology works is we work in the lab and generate new data. Does that make sense?
Yes.
And so that I think is I think what you're seeing the early days of the AI for science thing is the things that are easy for a frontier lab to do in-house, stuff like math and maybe some physics. What's coming is connecting that to the real world. And so this system is how at PNNL, they're able to connect AI and large compute into the real world. In their case, what they care about is the real world of these microbes that live in the soil. And what they're trying to understand is, hey, I've got a microbe in the soil sample that breaks down this toxin, for example. How? And that's phenotype, genotype. So in other words, the phenotype is it's breaking this thing down. The genotype is the DNA code that tells it to do that. And if you're like a generalist or a tech person, I'll just point out cells are the other coded discipline in addition to computers. They're like the only other one, right? Like this chair, there's no code inside of it that defines its shape and what it does, okay? But in every cell in your body, and the plants outside, and the microbes in this room, and the fish in the water out there, there is code that defines how they were assembled, how they operate. That's crazy, right? Like it's crazy to have physical things like that. And so this genotype-phenotype mapping is basically human beings getting to reverse engineer the 4 billion years of evolution that went into you, me, the fish, the birds, and learn how they work.
Fascinating. So in that, again, from what I'm understanding is you're observing how different microbes are breaking down different toxins.
Yep.
Maybe you're narrowing in on the ones that are particularly effective.
Yep.
And then you're trying to understand, reverse engineer, what is the code, right, of life.
Right, like you, for example, like the approach you might take is you might start to perturb different genes in that microbe, right? So you might isolate it and then make edits to its genome. And then you might look at the expression levels of genes based on those edits. You might look at the phenotype. Did it lose the ability to break down? Did it get better at breaking it down? And you can kind of start to work your way into understanding how it does what it does. That's like some of the things you might do. So perturbation and assays. Well, in order to do that, again, we're not living in computers. If you want to program DNA code, you have to do physical things. If you want to make an edit, you might use like a CRISPR system. If you want to add a gene, you would use DNA synthesis or DNA printing, where you go ATCGG, say what you want, and then in the lab, you can build that gene, and then that you would do that with a provider. They wouldn't do that on the system, but then that gene comes in and you got to stick it in the cell. That you would do, on the automation, for example. Does that make sense?
It does. So that project was a precursor to that announcement that you spoke of in December with Secretary Wright. And in that, you were awarded, it says, a $47 million contract to build the world's largest autonomous lab at PNNL. And it's called the Microbial Molecular Phenotyping Capability, or M2PC, which seems like generates rich, reproducible microbial and microbiome data. When I hear of microbiome, I think more of kind of the human health part of it. So what is different about this engagement?
Yeah, so what's different about that lab is the scale of it. Okay, so the 18s, can I give you an analogy from transportation to try to explain what we're doing here? So we say that's an autonomous lab. All right, so what the heck does that mean? Okay, all right, so I'm gonna give you an analogy from the autonomous car. So let me draw a graph for you here. All right, so on this axis is the level of automation. So on the y-axis. On the x-axis, this way, is the variability in the request from a user. All right, so in transportation, low variability, high amount of automation, that's the subway. You sit down, I swear to God, Nabil, it takes you to whatever, it takes you away, right? Totally automated transport. But you have to want to go to one of the stops on that subway line. It's not gonna take you to your house.
Right.
Low automation, high variability, that's a car. Put your hands on the wheel, foot on the pedal, and you can drive it straight to your front door. All right, so that's been the transportation system for the last 100 years. Until 4 or 5 years ago, if you've been around San Francisco, you get in a Waymo, and you're up here in this corner of the graph. It's as automated as a subway. You just sit down, but it takes you to your house. It has the variability of a car. And it's so surprising, because we've had 100 years of trying to automate transport and never being able to offer that amount of flexibility, that we give it a new word. We call it autonomy. We call it an autonomous car. And it isn't about, this is like a little bit of a confusion, it isn't about the human not controlling it, because you are telling it where to go. It's about the human being removed from the manual process of driving the car. Does that make sense? Okay, but still having the flexibility that used to require a human. Great, all right, so now let's talk about labs and what they're building at PNNL. All right, so low amount of flexibility, high amount of automation. We actually have those. They're called work cells, all right? And they're used, let's say you were Quest Diagnostics and you were running a bunch of blood tests every day. You would have a robot doing that for you. Why? Why not, right? So repeat, it's like a subway system. If you're a pharmaceutical lab and you're doing what's called high throughput screening, this is where you try a lot of chemicals and find one that makes this tumor cell die, and you hope then it works in humans. You might screen millions of chemicals, same test over and over again. That's already been automated. There's no humans doing that. All right, great. So that's your subway, that's your work cell. low automation, high amount of variability. That's the lab bench. Okay, this is what I did. I did my PhD at MIT, bioengineering. It's 5 years of moving liquids around by hand, okay, and working with different devices at the benchtop by hand. Put a sample on, hit the settings, okay? And so why is that so persistent, right? Like I talk to tech people like, automate it, right? And the reason it hasn't been automated is for the same reason the car hadn't been automated. It's too variable. The amount, like what you do every day as a scientist is different, not exactly different, but slightly different than the day before and very different from what you're doing 6 months ago. Does that make sense? All right, so there we've sat for, in our case, the last 40 years since kind of lab automation got going, until what you'll see in a minute and what PNNL ordered from us, which is an autonomous lab. And I'm trying to get up to this corner where you get the automation of a work cell. Like there's not a human in there. The experiment runs start to finish without a person, but you can request as the user the same kind of variability you would get from the bench, different experiments every day. So what's cool about that system, so for starters, when you're in a normal walk-in lab, like a manual lab, there's maybe 30 or 40 different pieces of equipment you have access to. Well, the first system I built for them only has 18 robots. So there's only 18 pieces of equipment. And so that means there's some experiment where you need equipment that's not on the system. 97, that's a lot more equipment. So what we're doing is in part just creating the full range of equipment you would need for the many different protocols you would normally do in your walk-in lab, in your manual lab. Does that make sense? For the kind of work they're doing. And by the way, it's like you're future-proofing it because it can do flexible stuff. I don't need to know exactly what you're doing.
Okay, so it's not just about microbiome type work. It could be
anything that uses that equipment, right? You know, and so that equipment that we've chosen, that they've chosen, to be fair, is the type of equipment that's been important to them for doing the kind of microbial work they do. So, but if you wanted to use that equipment for something different, the lab could certainly accommodate that. Right, and so that's what I think is really neat. I think we're doing that's fundamentally different at Ginkgo. That's like the new contribution to this whole AI for science and new contribution in lab automation is to go for the autonomous lab, like the Waymo. I want to give you the variability of the bench, and no one else is doing that.
Well, you mentioned that this announcement came, in a way at the same time as Project Genesis, which is a large initiative from the government, as you had alluded to earlier, around consolidating data sets and really trying to create kind of AI-first, scientific discovery. In reading up about it and kind of getting a sense of what the drivers were, one of them, I guess one fact that I learned recently is that one of our peers or competitors, depending on how you think about them in China, where something like there's a site called Global Data, which is a research site. They say estimate that Chinese innovation drugs accounted for 28% of large pharma innovator licensing deals. It says by 2024. This information is dated. I've even heard some numbers closer to 40%.
40%, yeah.
Okay. So one thing that I find interesting is
46% of new clinical trials.
Okay, well, there we go. So, China, for example, as an instance, isn't now just doing sort of like the easier, sort of maybe less sophisticated research work. They're now bringing drugs to a level of development of sophistication that American large pharmaceutical companies are themselves taking bets on them. So one option is, or one question is, how do we stay competitive in that environment? Seems like Project Genesis is a response, at least in part to that. Would you agree?
Yeah, so I had the opportunity to chair a National Security Commission on emerging biotech for 2 years. This was started 3.5 years ago. So I chaired it for the first 2 years, and then Senator Young from Indiana chairs it now, and it'll wrap up, I think, in the next few months or so. But it's a congressional commission, and 2 senators, 2 Congress people on it, and it was created to ensure America. What do we need to do to ensure the U.S. stays competitive in an important technology area? There was a similar commission like now 8 years ago for AI, and this one's for emerging biotech. Okay. And so we looked at this a lot in the commission, and I would say the way you described it is exactly right. There's been a huge shift. Like the backstory is that number of like acquired therapeutics from China 4 years ago was less than 5%. That's now 40. So what happened? 2 things. So one is regulatory. Chinese FDA changed how they did things maybe now 5-10 years ago, and that's kind of worked its way through the system, and it's just much faster to do a phase 1 trial in China. About 6 months, similarly in Australia.
Is that this implicit approval system?
It's basically like investigators in China can approve the trials Like it'd be like the, you know, like investigator-led trial works for their FDA for a drug, in any case. And so Australia has a system too, by the way, that also goes just as fast. So we don't even really need to totally copy the Chinese system. We just copy the Australia system. And I am hopeful that the US FDA will do that, like soon. And so that's one part. But that's not the whole story. And the story that reads on science broadly, and then biotech in particular, as I would say our most science-forward private sector industry. That's why like we do a lot of science where really it's just the government funding. Hey, I want to learn about this, and let's learn about space or whatever. And you know, maybe now space is becoming an industry, but for a lot of time it's just purely science for science sake, right? Which I think is important. But biotech and pharmaceuticals is an industry, but the drugs come from scientific discovery. Okay, so it is a very good bellwether for like what's happening with science broadly. Does that make sense? All right, so why are we losing it? And I think the other thing that changed in the last 15 years, basically, was it used to be the case that the US had the monopoly on the scientific talent, because it's actually quite hard to produce scientific talent. You need to be an advanced nation. You need to have people like willing to do a PhD and a postdoc, you know, like it's like an advanced degree thing with like the prospects are marginal, like, you know, like you're gonna become a professor. Like you have to believe that like this whole system can support that group of people, right? And so it's hard. And then you have to have the system. So first you gotta be willing to pay for it. Then you gotta have the schools and the da-da-da. And China built all that stuff over the last 20 years. And they just used to not have it. So we used to kind of by default be the only place that could do the science of biotechnology other than Europe. And that's just not true anymore. Does that make sense? And then here's the real kicker. They don't pay their scientists as much. And remember, how does science advance? At the lab bench. I cannot emphasize this enough. Like we can be smarter, but at the end of the day, what actually moves the field is the experimental work. So there is very much a component of science that looks roughly like variable light manufacturing. And variable light manufacturing, China is super good at economically. Like they have the people still willing to work for that, you know, at a certain cost basis to do manual variable work. Does that make sense? And so now if you have an educated enough person to do the manual variable work of science at the price point of China, they get twice as much research per dollar as us. So what the hell are we thinking, right? Like so we are at a point right now where I think we need to fundamentally rethink the way the United States doesn't just do biotechnology, but the way we do essentially laboratory, particularly manual lab bench laboratory science. The accelerators and things like that are different if it's like a big capital project, but like the manual work, all the fields that are moved forward on manual work, I don't see how we're competitive anymore. And so that's why I think the autonomy thing is such a big deal. I'm glad to see it's part of the Genesis mission. I think we're also leading on the frontier AI models side, so we'll have smarter ideas and we'll be better at working with those things. But it is not a freebie anymore, right? It was a freebie basically after the Soviet Union fell. We had like a freebie 40 years. Right? Back, remember the Russians, like the USSR was not bad at science, okay, right? Like that was a real, like, you know, that was real stuff. Yeah, exactly. We made DARPA because of Sputnik, right? Like that was the last time we had a peer competitor in science. Okay, it doesn't feel great. So I think we want to make sure that we change how we do it to be competitive. And I am starting to see that happening in DC, and there's awareness, and you know, there's a whole, obviously a bunch of new sheriffs down there. So like, let's see how it goes. But I think Mike Kratsios in particular in the OSTP is excellent, and he gets this stuff. So, and that's sort of the cabinet member in charge of science.
There's an interesting, you know, we're in the kind of dry dock area or the dock area of Boston.
Yeah, I got a working dry dock out the window right here.
That's right. And I feel like there's almost a theme of this reindustrialization of America, of trying to, you know, onshore again, you know, manufacturing, light industry, but also do that in a smart way, right? Given the talent pool that we have and the abilities that we have.
Yeah, could not agree more. So if you look at a company like, say, Hadrian, I don't know if you're not familiar with them, they're in LA. They basically do like high mix, lower volume manufacturing for the defense sector. So they're the ones making initially the prototypes and then some of the smaller scale manufacturing for Anduril, SpaceX, like all those like subcomponent parts and all these things. And it is the kind of things where we would previously get a lot of those parts from China. All right. And so they are figuring out how to do that in the United States. And they've made way more investments in automation, way more. So, and that allows them to bring in like a different type of employee who can kind of work with the automation rather than, oh, we're going to have skilled machinists. That doesn't really work, right? Those aren't jobs that can pencil versus the competition in China. So that's the problem. Like, yeah, sure, we could have it, then everything we make will just be more expensive. So we don't get to go back to 1950s manufacturing, and science needs to get out of 1950s lab bench, or else it's over. Just like we lost all the electronics and everything. Like, it doesn't compete, right? I'm just telling you, it doesn't compete. But the good fact is, like, look at the Waymo. What's happening with the AI models is it is starting to bring robotics into this variable work. Like obviously the other huge area of investment right now is all these humanoid robots and everything else. This is all part of the same theme, which is the US being able to automate the physical work that we previously couldn't automate technologically and bring it back from China.
So I think
all across the board. And I'm just the science angle on it, but a place like Hadrian is the manufacturing angle on it.
So, I think America's, sort of superpower as an organization or as a country has been its ability to iterate quickly, right? And hopefully learn our lessons and be able to stand these things up.
We are fast.
Yeah,
I would agree.
So, with that, there are growing pains of trying to work in these new ways. And so I'm curious about, as you're, learning as you go, as you build out your own lab here with PNNL, as they're building out this much larger rack system and it increases in complexity. I'm curious to explore some of the challenges that come with kind of scaling up. So, one of them that I have I've spoken to folks about, who've been involved with organizations like Emerald Cloud Labs and Stratios, these sort of cloud wet labs that also had this dream of being able to be
Max Hodak last week. Okay. He's the original founder of Transcriptic that became Stratios. Yeah.
Great. So the AWS of science, where you could not, the person in their dorm room could be able to run an experiment. Yeah. Some of the challenges have been like, for example, scientists maybe aren't educated in translating their science into code or into specific protocols that can be, placed on these machines. And there's the economics of just capital-intensive, machines and trying to maximize uptime and so forth. So, what are some of the challenges that you think we're going to need to solve just as a country as we take this path for science?
Yeah, that's a freaking awesome question. So and again, I would say the major thing, like the most distilled version of the technology problem was what I told you on the graph. It's like, get to the top right corner.
Get to the Waymo.
Yeah, and like even think about Waymo, right? So that boy, was their challenge hard, because the most dangerous thing I do every day is get in a car. So they had a huge bar, right? It was like worth nothing until it was safer, until they knew it could get safer than people. Yeah. It was worth $0. Right? And so
something like a decade, I think.
Yeah, I'm just telling you. But if that had never got safer than human drivers, it's now much safer than human drivers. If it never got safer, it's a goose egg. It's over. Does that make sense? Okay. We're not in exactly that bad of a spot. Okay, right? Like people, if I'm not as good at some of the things at the lab bench, someone doesn't die or something. Okay, right? So I can kind of glide in there a little more easily. But let's just be clear. That is the core technical challenge. It is managing the variability with total automation. There is no doubt that if I had a lab that ran 24/7, where I could compress all the instruments into just the space that the robots need to move stuff around, not the space it needs for humans to wander, and it was running with a very small number of people instead of many people inside of it, which reduces EHS requirements, safety stuff, everything else, that will be dramatically superior on a cost basis, a time basis, runs 24/7, not 5 days a week, 6 hours a day or whatever, like a normal lab. It is an obvious winner economically, competitively with China, however you want to look at it. So it's really a question of, can you actually do that? Does that make sense? Okay. So then once you believe that, which I deeply do, then the next question is, what makes that hard? Okay, right? And we can talk about Emerald Cloud and things like that, which is a little more on a business model side. But let's first talk about what makes it technically hard. Okay, so what makes it technically hard? All right. First off, you need to support many different pieces of equipment all in one system. Because and the best way to think about all this is always to look at how a manual lab works. All right, so don't compare to a subway. That's the mistake actually people in the pharma industry and others do, because when I say lab automation, they say
Liquid handlers.
works out. Yeah, exactly. They're like, I got this. I know about lab automation. Let me talk to you about the system that screens my antibodies or something. And I'm like, yes, I agree. It's a great subway system. I think it's wonderful. You should keep it. I'm not trying to shut the subway down in Cambridge. I'm trying to get rid of the drivers. Okay, right? Like I want us to all be riding in self-driving cars. I'm not actually trying to replace the thing that goes to the same place every day because a lot of people need to go there. That's fine. Keep it. Okay, right? So I want to compare to the by-hand lab. Well, let's talk about what a by-hand lab looks like. All right, so you might have a room. It's I don't know. Let's it's a 5,000 square foot room. There's 10 workstations for scientists to pipette at, and there's 60 different pieces of laboratory benchtop equipment in that room. Freezers, bench space, but then PCR machines and centrifuges and all the different stuff they need. And that's all in there available to every one of those, let's call it 30 scientists that work in there. All right, so that's a manual lab. I need to offer the same thing. What makes it hard to do that? First off, try putting 50 pieces of equipment on one setup. Okay, well now your little arm system, and it needs to be a little more complicated. You need a transport system too. You need rails and this, that, and everything else. Okay, so you gotta solve moving samples with high reliability among 50 devices. You also have to integrate 50 different devices. I can't have a person in there programming that thing. All those devices need to be controlled by my orchestration software that sits across the whole thing. So I gotta solve that problem. All right, so you need to handle all 50. Now people have done that in the work cell. Biosero, high-res, we've obviously done thermo. The talking to the devices can get solved. 50 devices, 100 devices, every time that's been tried to do as a work cell, it's like a Frankenstein's monster. Okay, right? And there's a bunch of these old stories. Oh, Lilly built this facility and 20 years ago it was a fail. Like everyone who's tried to put too many things on a old inflexible system, it eventually like kaputs. Okay, so you have to figure out how to do that scalably. Okay, number two, you need to be able to do lots of different things at once. Because it's not the case that when you go in that lab, you lock the door behind you and you say, lab in use, no one else can come in. Yeah. But that's how the work cell works. Okay, right? If you're using it, I can't use it.
It's a lot of parallel processing.
Scheduling, right? And so it's like you and I using the lab together, right? And we'll see this on the lab tour, but I'll show you. But we have an advanced scheduler to handle that. Okay, and then the third thing is it has to be easy. So, for example, if you want to do work at the lab bench, you don't have to write code. So you cannot write code to use the autonomous lab. Make sense? Okay. So that needs to get solved. Yeah. All right. And so those are the big problems that you have to get over in order to make this thing work up here. Right. And they're all, the good fact about them is they're all engineering problems. They're not science problems. There's not like some unknown thing I gotta discover about biology or something here. I just have to reduce these things to good practice, good UI for scientists, and have it be reliable. Okay, and I think they're all imminently doable. And we have made major headways on them, and then we can talk more about that. But the last point I'll make is the unique advantage we have at Ginkgo is we have two halves to our business. We do the automation and tools business where we sell these things to other people, but we also do, like we can talk cloud labs in a minute, but like we do sort of like research projects for other companies. So I have, deals with Novo Nordisk, Bayer Crop Science. We do like multi-year complex research work. So I have scientists here. The Ginkgo scientists are like the Waymo engineers 5 years ago in Palo Alto, not in the backseat of the Waymo, in the driver's seat, right? And they're there and they have their hands like right next to the wheel. And if it turns into a mailbox, they grab the wheel at the last second. Okay, that's my scientists on my autonomous lab today. I'm like, we're gonna try to hit this month 30 simultaneous protocols. Jason, that's gonna break the scheduler. Let's find out. It did break the scheduler. Great. Now the software team knows what breaks when you try to run 30 real science protocols at the same time, not some toy thing, and they fixed it. Yeah. Great. And what's the next problem, right? And so by having like our own people here to like dog food it, we are moving dramatically faster than anybody else towards this goal. So I think we absolutely get there first, but those are the hard problems to solve. Many instruments on one system, parallelization, and ease of use by the scientists. Those are the three most important ones. There's a fourth, which is sort of like, how do you get the atoms onto the system? Okay, right? Like that can't be as much of a project as using the system. So you kind of want to make that easier too. In other words, like how do you get the reagents on and your cell line on? Like how does that feel again to the scientist user? And that's it. We just knock those down. And feel like that's it, and then we're done. We're done with the benches, and science can actually start to move scalably, right? Because I'll make one last rant. Like I said, as a project Jurassic Park, I went to MIT as an undergrad, and I was like, I'm going to go learn to do genetic engineering. Like freshman year summer, I'm like, I'm not going home. Forget Florida. I'm going to stay at MIT, and I'm going to work in a biology research lab for the summer, which I did. It's got Herman Eisen's lab at MIT Biology. He's passed away now, but it's a cancer lab, and it was like, and my project, I was given like, I was working for this like postdoc, and he was really nice, and he was like, I'm going to give you a project that's like a classic start biotech project. You're going to move a gene, okay, and you're gonna take a gene, you're gonna PCR it up, and you're gonna put it into E. coli bacteria. It's like cloning. This is like what Genentech, and Breuer and people did in 1978. It's like the beginning of biotech. Swear to God, I spent that whole summer trying to clone one gene. And I was like, oh no, Jurassic Park's not gonna happen, right? Like it was devastating. Like I was devastated. And I was like, maybe I just suck. And so, I took I was taking the biology lab classes, and the second year and third year, every year I did research in the summer. And even by my senior year, I was obviously better experimentalist, but I was like, wow, this is hard, And I was fortunate to meet, I ended up working for Drew Endy and this fellow Tom Knight who started the company with me here, and my other folks I did grad school with. But like, I found a crew who were like, you're right, Jason, it's totally hard, but we have a plan to fix it. And I was like, okay, I can get behind that. But we don't get to do any of the cool stuff. Like Ginkgo's missions for the last 17 years has been make biology easier to engineer. This is the blocker. The lab bench is the blocker to making biotech like tech. You have to stop being manual. Computers are called computers because they used to be a job to do the slide rules. Right. And we automated it. Yeah. And then from there, it just ran for 90 years. Mm-hmm. If we can make the process of programming biology fully automated end to end, then we can start working on cost reduction from there and scale. Mm-hmm. And I swear to God, it'll end up like the tech industry, but bigger. Mm-hmm. Okay, because it's in the land of atoms, mm-hmm. which is much bigger markets. Mm-hmm. But we gotta get over it. We gotta stop the bench. So does that make sense?
It does, and you had mentioned earlier, the business side of it. I want to get into it later.
Yeah, let's definitely do business models. Yeah,
after the lab tour.
It's Sloaney, right?
That's right.
This
is my comfort zone.
Yeah. Let's go.
But I want to touch on that quickly because it relates to your recent work with OpenAI.
Yeah.
So you had an announcement with OpenAI around really, it seems like reducing the cost of cell-free protein synthesis.
Yep.
And we'll actually get to see the lab where sort of the AI model was mated with the autonomous lab and where the work got to be done. And I feel like there's an interesting, almost kind of back and forth relationship with when you go into autonomy, which is there's, from the Nature article that I read and so forth about it, a real emphasis on reducing costs in order to do science and be able to do science at scale and be able to look at a much larger, maybe like problem space in order to find interesting solutions. When you do work in the real world, it requires reagents and precursor chemicals and all sorts of very expensive stuff. At the same time, right, it seems like the North Star for this work that you did was to reduce those costs. So in some ways, it's like to do the work requires maybe more cost than usual, but then as you do it, maybe you find efficiencies that otherwise wouldn't have been found. So tell me more about that. It seems like a really interesting dynamic here.
Yeah, so that's a very good insight you had there. Actually, that's one of the reasons we picked that as a target for the project, because we thought it was sort of like AI helping itself. Because one of the big challenges, as you're pointing out, and this is again why the frontier labs are starting with math and physics, is that's free, right? Why did DeepMind start with chess, right? Like, well, you can play games of chess for nothing, right? And you're on 1 million games of chess on a processor, right, for nothing. And so, yeah, it uses power, but like comparative to using atoms and building things, it's cheap. So absolutely, right? Like now, to be fair, again, the dominant cost today, I cannot emphasize enough in doing the atoms work of biotechnology is humans and physical space, not the reagents. The reagent budget, even at a highly automated place like Ginkgo, like back when I wasn't on the autonomous lab and we were just doing like walk-up automation and work cells, it was like sub 7% of the budget. It's just dominate, it's just so expensive given relative to the throughput of the people. Okay, so let's just be clear. The actual first problem is remove the vast amounts of like overhead for inefficient lab space, low utilization devices, and humans moving stuff around all that, and let the humans think and not do that manual labor. Okay, that is step one. After you've completed that step, it's still going to be expensive compared to playing chess. But at least it'll be less expensive than doing it at the bench. Does that make sense? Okay, and then now we're into the era of, oh, it's all running autonomous and it's all about the reagent costs. Why don't we reduce the cost of the reagents? And so that was the project we did with OpenAI. We're like, one of the best fits, we think, for a lot of the AI for bio stuff right now is in just protein design. It's a tractable problem. Proteins are not the infinite complexity of a whole cell. And so starting with AlphaFold at Google, and like Demis won a Nobel Prize for this, but like on to newer models, newer versions of AlphaFold, but companies like Chai Bio and others are basically making models to design proteins against therapeutic targets. Great. How do you make the protein? Well, you can obviously put it into a cell, like a CHO cell, but when you do manufacturing, it's typically done this way. But in the lab to test many proteins, it's often done in a cell-free mix, at least for these AI guys especially, because it's much faster to make the proteins. You're not waiting around to like grow a cell line up. So you add your DNA to what cell-free basically means. I grew up some cells, and then I popped them open, took their guts, put it in a tube, and then when I add DNA to it, remember high school biology, DNA, RNA, protein, it turns into protein. All right, and so you can change what else is in the mix. You can add endogenous extra chemicals to the mix that aren't just the guts of the cell. Depending on what you add, you get more protein out. The things you add cost money, but you know how much money they cost. So you could say a science project would be, what is the most optimal set of things you can add that give you the most protein for the least dollars? Okay, so protein per dollar produced. And that was what we set as the metric. We're like, GPT-5, your job is to put the right things in this mix that give us the most protein per dollar, all right? And the thing we got lucky on, we actually already chosen this as the project. And then Mike Jewett at Stanford, who's like the world's leading like cell-free, in my opinion, protein synthesis guy, he published a paper that basically benchmarked a bunch of the different current mixes on price. And then his student did a ton of experiments, ultimately not as many as we did. We'll talk about that in a second. But and beat state of the art by 90%. And so there was a new state of the art as of August. And we're like, let's beat that. Okay, and then we ran the experiment. We at first we let GPT-5 basically design we only gave it access to like its own brain. So it didn't have a computer, it couldn't read the internet, only on whatever had been trained on originally. All right, and this was actually GPT-5. We're on 5.4 now, but we started at the experiment, it was 5, and so we kept it 5 the whole time. All right, so we're like, okay, figure it out with what you know. And it designed experiment. Now we had a rule. This is actually kind of useful because like for doing AI for science, I think this is the biggest experimental AI for science result that has happened so far. Okay, and so I'll give you some tips and tricks for people that are interested in bringing AI into science. So one of the things we did was we had what's called a Pydantic model, and this is open source, you can download it, our model, but it basically is a set of formal rules that in order for the model's experimental design to end up on the autonomous lab at Ginkgo, it has to pass these tests. So there's a 384 well plate. I can show you what one of these looks like, but there's like 384 little wells that you can add liquid to. So each one of those is an experiment. So a few things are in the rules. 1, you got to design 384. Don't waste any wells. 2, you can only add a certain volume of liquid. Otherwise it overflows. Or even at a certain level below overflowing, at the speed we move the things, it could slosh or something. So you have that as a rule, okay? So you set these rules. And also you need to reserve these 2 columns for controls, because we're going to publish this and we're going to need controls. And also you got to do replicates. So each one of your designs, you got to put it in 4 times in a row the same so that we have replicates. So we set those rules. But then within those rules, it's all up to you, GPT-5. You could just tell it to fill it with water, and we'll fill it with water. Okay, whatever stupid idea you have, we'll try. Does that make sense? All right. So first off, we didn't even know, would it be able to design an experiment that like express protein at all, right? Like, would it just do something crazy and ruin it or something, right? And so out of 500 of these 384 well plates that we ran, only 2 were like fill it with water, like where it really was like messed up. And partially, one of them, it was in part because we had the pedantic model, we missed something. And at one point, in order to try to get more reagents in, because we had that cap on volume, it added some reagents at a negative volume, which is, physically impossible, but hey, clever. And so we had to make a rule, you can't, and so like it messed up too. But 498 plates, in my opinion, were fine experiments to run. In other words, they would contribute some data back that it would learn something from. Does that make sense? All right. So we let it do that. And then for 2 rounds, we said, you don't get any information. And so it started off, I forget what it was 10 or 20 times worse than the benchmark with the first ideas. It got a little better and a little better. But it wasn't like dropping like a rock, but it was improving. So it was able to like look at the last round of experiments. And then when it designed the next round, it was better, not worse. So that's already interesting, right? Like these are things that we weren't sure, right? And then we said, all right, well, let's let it have everything a normal scientist would be if I gave a project to. You could go read the literature. Here's Mike's paper from Stanford, tells you like the latest and greatest. Here's a computer. You want to write code, here's Python compiler, like go to town. And then on the very next design, it beat state of the art. Okay, and then in the next 2 designs after that, we were able to beat state of the art by 40%. So pretty neat. Now, a couple things were like unfair to Mike. We did more experiments than they did. So was it smarter or did it just have more horsepower? Mike didn't have an autonomous lab. He had a really good grad student. They, right, who did a lot of experiments, right? But GPT-5 had access to robotics infrastructure. Right. Okay, so I think we don't yet know, like, is it better than a scientist at doing the same thing a scientist is doing? Sure. But we do know it can do the work of an experimental scientist, which we did not know before we started the experiment. Does that make sense? And we know it can do it at a level that's like, not even just like the beginning stuff where we showed, it can learn and do whatever, that it could read a current state-of-the-art paper and do the experiments that improve on the state-of-the-art. Like that's a big deal. Now, this isn't like cure cancer, it wasn't the most creative work. This is the kind of work that you would describe a little more as like exploring an experimental space. But hell, that's a lot. Like back to my point earlier, I think the Einsteins are the rarity of science, who are sort of stewing and coming up with a theory and a breakthrough. The majority of science is testing the real world. And just being logical about what you see, and then being careful about the experiments you run, and then sharing your results. It did all that. Yeah. So that means we can run these things. Like if we want to think about the US running science, why aren't we spinning up 10,000 agents, working on a particular science problem, all with access to autonomous labs, doing different experimental hypotheses, and work on it?
What advice would you have for, the frontier labs of the world, and maybe in your conversation with OpenAI, as they're looking to make sure that their next release of a model is more effective in having the kind of intelligence to come up with better hypotheses scientifically? Is it running more work with you and being able to use that almost as a version of like reinforcement learning with human feedback, or I guess autonomous robotic feedback? Is it a different version of that? What do you think will get to the next level of intelligence?
I think for the task of doing science, it will help to get feedback on science. Like I think there's one way to think about this is in terms of like careful logic, and I don't know, the way I think about science is like it's like formalized human curiosity. It's like where we do the stuff that we're like curious about. Like business is like that a little bit, but not so much. Like business is more about competition. Okay, right? Like we're playing a game of chess against each other, right? And strategy and this, that. There's like, but that's a certain kind of intelligence. It is, like without question, right? Some people are much better business people and much better chess players. But great scientists are like experts in curiosity. And I think that kind of intelligence is maybe the most important kind of human intelligence. Right? Like what got us out of the trees, right? Like plenty of other ants compete. Ants go to war, right? Like like they're the only species that goes to war, but they compete and go to war. I'm sure they have some strategy to it. But they don't invent. So I think this particular line is is something that frontier models should focus on. And I think you'll get some of it from math and physics, but there might be some pathology to the things that are purely theoretical. Most of what humans have invented over the years, obviously, is in the land of atoms.
Well, speaking of the land of atoms and where theory meets practice, we have an awesome lab here just steps away from us.
Yeah.
Would love to grab a tour with you.
Let's do it. Yeah.
Okay.
Awesome.
Great. Good.
Yeah.
We're all suited up here,
Yeah.
and we're about to enter what exactly?
Okay, so you're about to get a tour of the world's largest autonomous lab.
Okay.
We call it Nebula.
Nebula.
And it's 52 of our rack automation carts, so 52 robotic arms
Okay.
connected to about 70 pieces of laboratory equipment.
Okay. RAC is an acronym RAC. Is that right? What does that stand for?
Reconfigurable automation cart.
Reconfigurable automation cart.
Yeah.
Okay. There we go. All right. Let's check it out.
Yeah, let's do it. Okay, so this one actually says touch me. We had this at SLAS.
Oh, nice.
Yeah, and then, so one thing that's kind of funny, right? So we had like 5 of these set up for this laboratory automation conference.
Yeah.
And then when it was done, we just brought them over here and installed them in a day. So this is like one of these things, because we've standardized, like our product is actually this cart.
Yeah.
It allows us to very quickly expand these systems. That's very different than traditional automation.
Okay. Can you walk us through the components of the cart? What's happening up top and at the bottom? And
Absolutely, yeah. And so here, come over here and take a look. Back to that like by hand lab we talked about.
Yeah.
So if you were setting up a new lab for some scientists,
yeah.
you would ask them not what protocol they're doing. That's what you would do if you were building the subway.
Yeah.
Right. You would ask them, what kind of work are you going to do and what equipment do you need?
That's right.
And then you're going to fill that lab like we talked about for 30 people with 50 different pieces of equipment.
Sure.
Okay. This is that 50 equipment lab, except without people with robots.
Yeah.
And my unit of design is a device. Okay, so you told me you needed this device. Well, great. When your scientist interacts with this device at the lab bench, they can do 3 things really well. 1, they can put a sample onto the device. Sounds simple, right? But they don't screw it up. They don't drop it or smash it or something like that. They just get it on there every freaking time.
Yeah.
Right? Does that make sense? So that's what we've done with these. You can see this one running here in the 6-axis robotic arm. It just put that plate where it was supposed to go. And it picked it up off one of these delivery carts.
Okay.
All right. And you'll notice that the track is part of the metal frame here. That means this arm and that track and this device are always in the same place in 3-dimensional space. Okay. And so that means that's the trash rack.
Oh, there we go.
Throw stuff away.
Okay.
Yeah, you'll see that in a second. But okay. So you're able to always get that right.
Okay.
Second, in a by-hand lab, you can walk to the other side of this lab with your feet
Yeah.
and bring a sample to another device, right?
Yeah. Yeah.
All right. Every device you see on this setup are all connected by this track. And I can, with software, send this plate here onto that cart and send it all the way over to the device in the corner.
Okay.
Just like you could walk over there.
Sure. Yeah.
So that's the second thing.
Yeah.
Number 2, transport. Third, this has some little touchscreen on it.
Yeah.
You can set the settings because depending on what experiment you're doing, you need different settings on this particular piece of benchtop equipment.
Sure.
Okay. So underneath and.
How would I access that if I
Yeah.
wanted to?
So you'll access it. You'll actually access it through our software.
Okay.
But just so you know, like what's going on behind the scenes,
Oh, wow.
we have our electronics box and we have drivers that we write for each one of these devices. So we're able to power cycle this. We're able to set the settings to whatever we want, all via the software.
Okay.
Does that make sense?
It does.
So now you can do the same things you could with a normal device once it's inside this hardware and software wrapper of the rack. Now it's available to you just like it would have been at the bench.
Okay.
And you connect it with its friends and now you can do protocols across them.
Okay.
All right.
So it's interesting you use the word wrapper because I thought of that from a software perspective.
Got it.
So this is a hardware wrapper in a way.
It's a hardware and software wrapper around a benchtop lab device.
Huh.
Does that make sense?
Is there, I guess, what inspired this wrapper concept?
We had been building a lot of subway work cells and trying to change them and realizing that making a change to that took 3 to 6 months.
Okay.
And so the flexibility that the scientists needed, hey, I'm doing a different experiment today.
Yeah.
I can't wait 2 months to reconfigure your work cell.
Sure.
But with these, we could just change it in the software and it would magically work. So we were trying to solve that problem for ourselves. And that's why we built this hardware. We built the first generation of this 12 years ago or something.
Yeah.
This is the newer generation, came out 2 years ago.
Okay.
The software is improving all the time. And I can show you one other. Do you have a question? Go ahead.
Yeah, I was going to say, like, I would imagine a lot of debate went in because you want probably the smallest minimal size, but large enough to fit a variety of different equipment.
Yeah.
I do see that you have two different
Right
size
here we can
models
see the
here.
3 sizes.
Oh,
It's
okay.
like sort of small, medium, and large.
Okay.
Trash rack there. And that's exactly what you said.
Okay.
To manage different types of lab equipment.
Sure.
And there's even little tweaks that we do inside. I can show you that are oriented around that particular piece of equipment. So every time we bring online a new equipment, we do the work to get it working really well inside the rack.
Okay.
One-time work, it takes about a month, and then that equipment is now part of the system. Yeah.
So to get back to the Waymo analogy, so right now I've taken some Waymos in San Francisco. They're Jaguar, I think F-type or I-type sort of electric cars with a lot of different sensors bolted on and software integrated. The idea is, as we're starting to see with like Zoox and so forth, you build machines from the ground up.
Yeah.
I'm curious, 20 years from now, as we look at autonomous labs, I imagine this wrapper is going to disappear, right? What do you think it might look like?
Okay. I'll tell you what I think will happen in the 20 years out. There's gonna be some crazier stuff.
Okay.
Like it'll be more like sequencing, but in the near end, I think, let me show you something absurd.
Okay.
This device, multi-flow, great device, right? I guess, not really, but it's fine. See that touch screen on it?
Yeah.
Nabil, why is there a touch screen on a device that's interacted with a 6-axis robotic arm and controlled by an electronic box and software? What is the point? And the reason is because this machine was designed to be an island on a bench. And so it has to have on-device software that is very fragile and annoying, and on-device controls that are annoying and error-prone.
Yeah.
Right? You would rebuild this completely in the context of we're not using it as a walk-up lab, but rather we're using it as an autonomous lab. Does that make sense?
Yes.
So once autonomous labs become everywhere, then you will see a rebuild of the equipment.
Okay.
That's like Gen 1.
Sure.
And then Gen 2 is you're going all the way to stuff like we've seen with sequencing, where you don't even really think of interacting with that unit operation like you would have at the bench.
Okay.
Like sequencing went from, we're going to automate what people were doing at the bench. That was the beginning of sequencing.
Yeah.
To then into, we're going to treat it more like inkjet printing. And then so we're going to treat it more like microelectronics.
Yeah.
But that was over time. Does that make sense? I think we'll have a similar experience with the general set of lab work.
For sure.
Once we solve total automation of the work in the first place.
That's true. Interesting. Okay.
Okay, but let me give you another thing about a lab, right? So if like one of the devices, let's say the PCR machine in your lab breaks, you do not go to the front door of the lab, put up a sign that says lab closed.
Yeah.
No, you and your other 30 scientists using those 50 pieces of equipment just use 49 for the day, and one is down.
Yeah.
Same idea here. You push this blue button, just taking this device offline. I can now open this. I can pull this out. I can change in this case, this is how you would service this device. You maybe like replace this roll. And if that track in the front, that keeps working. But if there was a protocol that was supposed to get their plate picked up with that arm and put on this device, it will not do that. It'll go to a different version of this device somewhere else or bonus.
Okay.
Okay.
Interesting. This is a sealer, it seems like, or a plate sealer?
Yes, this is
okay.
And then you push that close. Back online.
Look at that.
Now the scheduler knows that you can now send things to this again.
Sure.
So that is much closer to the experience of, hey, the PCR machine is broken this morning, don't use it.
I see. That makes sense. This seems to be, I'm curious about, this transport system.
Yeah.
How often does this, get stuck, or you need to bottleneck things? Yeah.
Yeah, so it is so again, if you think about how a normal lab, again, manual lab,
For sure.
with 30 scientists in it doing their work, looks, you don't see them sprinting around the lab all the time, bringing samples everywhere. That's not what's happening.
Yeah.
Right. So what ends up happening is samples spend a lot of time on different devices as they work their way through a protocol.
For sure.
Okay, I'm on the PCR machine for 30 minutes, and then I'm going over here. And so the actual times that you need for a single protocol to use for transport system is a small fraction often of the total time of the protocol.
I see.
So it ends up being much less of a bottleneck than you might imagine.
Interesting.
Yeah.
Okay.
And you can also, things don't get really stuck behind anything, because the only time you would have to wait, even if you were right behind another puck, is the time it takes for it to take it off.
I see.
And then it's gone. Right? You're not leaving the puck there while the device is in use. You're going to bring another puck over just in time to pick up the sample when it's done. So the track is mostly, as you see, clear.
Yeah.
And then you have little cutoffs and stuff. So if you could be a little smart.
Sure.
And we're always we're working to improve the transportation algorithm just so it's it's faster, but like it's not today a big blocker.
Okay. So going back to just the engineering of this facility, and I've heard this reference to automating like car manufacturing and so forth, is there's the manufacturing that happens on the floor, but in a way there's the building that supports all of the robots in their manufacturing, right? The system for the system. So tell me more about kind of, again, the build out here. I see power in the ceiling. What are the kind of considerations as one is building a building that's automation for autonomy first?
Autonomous, friendly, So first off, you don't really need that much different than you would have in a traditional lab. Like if this is like house gas and air, right? Like it's the normal things you would have in a laboratory. In fact, this room was built not for this system. It's been here. We've got in this room for, I don't know, 4 years, 5 years or something. Since we first built it out.
Okay.
It was built for Ginkgo, knowing we were going to use certain amounts of walk-up automation and other stuff. And then we always had a design principle that we thought our lab would change. So we always built our rooms like big open footprint and put our benches on wheels.
Sure.
So that would be my I think the main decision is not to build like casework like built in.
Okay.
You've got it on wheels. Room, and then if you're going to have benches, have them on wheels. And then like what we're doing with this system, it's 52 racks right now. By May, it's going to be 100. So we're going to wheel those benches away. And we're going to build another loop and fill in the other half of this one.
Okay, there we go.
And so that is enabled by the fact that it's easy for me to push those out of the way.
Okay.
And then overhead utilities, I think is smart even in traditional labs at this point. You're seeing that more and more frequently.
Okay.
So I think that's almost becoming a standard.
Okay.
Yeah.
So we talked about this really interesting collaboration with OpenAI. This happened here, right, within this lab, as I understand it. I'm curious, like, how did OpenAI, I guess, how did that communication work? Were they, directly accessing these labs? Were they working in coordination with you on your systems? Like, tell me more about that interaction or communication method.
Yeah, so the basic interface was like JSON files, more or less.
Okay.
But like we were sending, they would send us remember I mentioned earlier, there's a certain number of the wells on 384 well plate.
Yep.
As long as it passed the Pydantic model, the GPT-5 could set the contents.
Sure.
So they would send over that list.
Okay.
These contents, these wells, and we had a protocol that was like, in this case, by the way, we really varied the contents, not the protocol itself.
Okay.
That's another thing I think would be really exciting to let the models try.
Yeah.
Hey, maybe try one of the other instruments on here would have been better to produce protein. I have no idea. Like, have at it, right?
Yeah.
So there's sort of a question of like, what level of abstraction is the model controlling? In the case of this experiment, it was controlling the reagents.
Sure.
And like the composition of the reaction conditions. Does that make sense?
Yeah.
So then it came as a protocol, but the one we had agreed on.
Yeah.
With a set of samples that had passed the Pydantic model, now would come into our software. That would then get translated into the protocol running, like basically the broker software on our system would accept that.
Sure.
And then we would run it, and what comes out at the end of that is a bunch of data. And we would send that back to OpenAI.
Okay.
And so they would get back the protein production level is the main thing they want to know for each well, and then any errors.
Great.
Something went sideways, that happens sometimes. The echo didn't shoot the droplets, an acoustic liquid handler that often will fail in some of the things. Like a couple of the wells didn't get what you planned.
Yeah.
But we know what they got.
Yeah.
So we give you the update on what's in there. Is that's all contained in the data.
Okay.
Does that make sense? They get all that back.
Yeah.
And then they come back with new designs after they've run some training with the model and had it look at the information.
Okay. So from JSON files to data outputs from your lab.
Yeah.
That's it.
Yeah.
Okay. I see behind us, there's what looks like a scheduler here. Can we? Yeah.
Yes, this is an important piece of the puzzle. Okay, so the other thing that happens in a normal by hand lab, I know, like a broken record on this, but I cannot emphasize how much, like, I gave that subway car analogy, right?
Yeah.
Miles traveled in the US anyway, but it's not too much different in Europe, between cars and trucks versus subways and trains. It's cars and trucks by humans. Miles traveled.
Miles traveled, like in total?
Yeah.
Oh.
What fraction are cars and trucks versus subways?
Oh man. I mean, I feel like cars and trucks would be a lot more than subways.
99%.
Okay. All right.
99% is cars and trucks.
Wow.
Because, and it's not like we don't know about subways and trains and rails.
Yeah.
It's because we need flexibility in transportation.
Yeah.
Right? And so I have had heads of R&D for a couple of pharma companies over, and I asked the same question when it comes to research budget for the subway automation, like work cells,
Sure.
versus the lab bin. Not development, not clinical trials, research equipment.
Okay. So a lot more for the lab.
99%.
Okay. All right.
If you walk around, you don't see any robots in these places. So the reason I'm a broken record on compare it to the by-hand lab is that is what we're spending 99% of our research dollars on.
Right.
Not the high throughput screening facility.
That's right.
That's only getting 1%.
Got it.
Does that make sense?
Yeah.
Okay, so in that lab, the other thing that is absolutely amazing about it is that 30 scientists all share at the same time. And they're able to use the same set of equipment.
Yeah.
So like I have my equipment, you have yours.
Yeah.
We have that for our pipettes, but like for the benchtop equipment, we share it.
Yeah.
And so we agree, you tell me, hey, I'm using the heat block in the morning, and I'm like, great, I won't use it until the afternoon.
Sure.
All right. That is what this is doing. So this is our scheduler. Each row on that is one of these devices.
Yep.
Like there's the trash rack. The one over there, there was a trash away, my favorite. Okay. And then here it goes down, you can't see it, but are all the different protocols submitted by, you can see the name of the scientist that submits it.
Okay.
And there's 36 currently running.
Okay.
So those are protocols submitted today by scientists onto the system. That's like the plates we saw moving around earlier. That was one of these things. This is now on the schedule, the orange line.
Sure.
And then for a given row, what you'll notice is that there's different colors.
Yeah.
They don't overlap with each other.
Okay.
Because the colors are the protocols.
Yeah.
And they both can't use the same device at the same time. So what you want is alternating colors on a device so that, it's done with it? Great. The blue protocol is done. Now the red protocol can use the PCR machine.
Fascinating.
And then when it's done, the green protocol can use it. And so if you submit a new job in with our software right now, it will check the timing of your protocol because you specify how long between steps you'll allow for because maybe it's time sensitive for something, maybe not for another step. And then if they can fit you into, now you can see the whole protocol list. And then if you can fit you into that, it'll fit you in. Does that make sense? And that is unique today in lab automation. No one has pulled off this level of parallelization. Like to have 36 protocols that were submitted today, we didn't even know what they were going to be.
Yeah.
And having them all running in parallel in a system.
Yeah.
There's no subway, there's no work cell that does that.
You know, I went to a quantum computing conference a few years ago, and they talked about the traveling salesman problem.
Like the salesman suitcase or the known OR.
Or traveling research aid.
It literally is. We had to do a lot of work to get the scheduler to be fast.
I was going to say, it gave me an appreciation for the incredible complexity that can come from trying to do those kinds of optimizations.
100%.
I'm curious, like what unlocks maybe have happened as you've been iterating on this, or do you foresee in the future? I guess, and is it just like a question of mathematical sort of, breakthroughs that enable that kind of coordination?
Different thing. So this is, again, I mentioned this a little earlier, but like the great thing about Ginkgo is we have our science research services business.
Sure.
So we're doing science here.
Yeah.
And we've historically done it in a bunch of other labs we walk by. There are a bunch of benches and walk up and work cell automation.
Yeah.
And I'm basically decommissioning those labs and moving everything onto this system as it grows. As we have like the equipment needed for the scientists to do their jobs, and I want them using this. All right. Again, this is a little bit like the Waymo engineers, right? Like holding the wheel. Okay. So I was like, great. I want us to have. This was like 4 or 5 months ago. I was like, I want to get to 30 plus protocols. Yeah. Like it's going to break the scheduler. Like, great. Let's see. It did break the scheduler, right? So you put 30 on, and like then suddenly things were taking like 6 hours in the queue and all this stuff, even though the equipment was available. It was because of the whatever salesman, not that traveling, but the other problem. And so, we had to do a bunch of work on that to figure out, well, what trade-offs would we accept?
Sure.
Right? And that was all really just pure software. But then we have other problems. Like, once you solve that, then you have, whatever, like we have a bunch of current issues, but like we have device issues even.
Yeah.
And you're like, okay, how do I debottleneck that device? What about getting samples onto the system? That's a slowdown. So we'll just hit things.
Yeah.
And my goal with this system here is I'll hit them all so that Merck doesn't indicate I don't need to. Okay, right? Like I will show that you can have 100 devices on a system here before I install the PNNL system in 97. So we'll have it in May. They're not going to have that system. They're waiting. They're building a building. I'll say 2 years.
Yeah.
Okay. So I'll have already shown it all.
You'll pressure test it. You'll make sure.
I'll bring everything first.
Yeah.
And so that's a real. We're again, Ginkgo's really unique in that regard. Like the hardware company is.
Yeah.
They're on the labs. So we're really seeing a lot of this stuff break for the first time.
Okay.
That makes sense.
Yeah.
And things break constantly. Is the short answer.
All right. So what else are we looking at?
So this is like the moral equivalent of the shelves above your bench. Okay,
Yeah.
it's room temperature,
Yep.
storage for your reagents and so forth. And
Yep.
we'll go down in a minute. You'll see even like, oh, actually this one has it. Your plastic tips, like the things you're consuming
Yep.
as you do your work, they're also on here.
Okay.
Okay, right? And so when this device, this is like a liquid handler, a Bravo liquid handler.
Yeah.
It's got some of those tips. You see the purple top?
Yeah.
The way they got there was the arm took them off here, put them on the little track, and it moved it over 3 feet and put it on that. As opposed to I open this up and I put the tips there.
I see.
Absolutely not.
Okay.
Right? Because that's an introduction of error.
Yeah.
Right now, humans got to be there to remember to do that. No. The human should just be loading things that are buffered,
Sure.
and then all of the stuff that involves bringing something just in time
Yeah.
and clearing things and trashing them
Yeah.
should all be done controlled by the software on the system. That's a design principle for an autonomous lab. Does that make sense?
It does. I'm wondering if there's any kind of like maybe unexpected benefit or maybe expected of, I mean, these aren't hermetically sealed, but I imagine fewer encounters with humans can help reduce contamination. Is that?
Yes, these are HEPA filter, positive pressure.
Okay.
We do mammalian work on here.
Okay.
One of our customers has been doing mammalian work on their system for 3 years.
Okay.
With no contamination events. And I think one of the big reasons is the people. So another open question is, could you do bacterial and mammalian on the same system? So that would be a major no-no at benches.
Sure.
And I'm like, let's try. So we've been running mammalian plates just like as contamination detectors. Mammalian cells, just floating them around while we've been doing bacterial work to see. So far, nothing.
Okay.
And I think the reason why is the source of the contamination.
Yeah. We're looking at it. Yeah. Yeah.
Way less. I think this is going to be like a dramatic improvement in that.
Is there a cold storage version equivalent of
Up
that?
here.
Okay.
Yeah. We got freezers, we got fridges. Yep.
Okay.
And we get these from third-party vendors.
Okay.
Again, we're integrating that into our ARM system, but there are already automated fridges, freezers, even -80 freezers that you can get.
How does a robot access it? Is there a door that opens at the robot?
Oh yeah, right here. Yeah, you'll be able to, well, it's not, if one opens, you'll be able to see better, but you can see here, there's like a little thing that slides over, and then it would reach in with its arm and get a plate out of a certain location, and then make it available to put it on that buffer that buffer seat, and then our arm would pick it up.
Okay.
So it's like an arm talking to an arm.
Wow.
Yeah.
Fascinating. Very cool.
Well, that works right. Well, there's a few different companies that make these types of things.
Okay. I'm sure the lab automation vendors themselves have a field day coming in here and just seeing how their machines are in this ecosystem.
Yeah, people really like, I mean, it's world unique right now. I mean, as we're going, some like the PNNL system would be an equivalent scale, but mostly when we're selling systems to customers right now, we're selling them opposite work sets. So we're building like smaller ones that are mostly meant to do one thing, but at least they know they could expand it in the future to an autonomous lab.
For sure.
I'm hopeful as I show more and more as possible on this, I'll have a few of the large biopharmas and others just buy whole autonomous lab. Hey, go ahead, guys, go. Sorry. So, but that's, right now, that's like, they're using their benches to do that work.
Right.
So I got to show them I can do what they could have done at their bench.
Got it.
Make sense?
Yeah.
Yeah. So that's where we are today.
All right.
Yeah.
Got more storage here.
So that's kind of the storage row. I'll show you another thing that's pretty interesting. So we talked a little bit before about the replacing, like the making the devices more automation first.
Yeah.
So we come here. So this device, this is a sealer. So you might have noticed when we had those plates in the storage rack over there on the room temp one, they were all sealed.
Okay.
That's because if you leave it open in the air, it evaporates. Everything in biology is liquid, right? Liquid space.
Yeah. The clock is always ticking.
Yeah, it'll evaporate behind you, right?
Yeah.
And so the there you go. Here you go. You can continue to see them. Okay. Their thing. So this is the device over here that does the sealing of those plates.
Okay.
It's a very finicky device. And the way it works is you have pressure coming into the back.
Okay.
That pressurizes it. You use pressure to push the seal down on the plate. All right, there's air pressure behind this.
I think my boba place has something like that. Yeah.
Not too wrong. Okay. It's very sensitive if your house air dips a little bit.
Okay.
And then it doesn't seal well, and you have a failed seal. It's really annoying. Okay. So what we did, see that little black tank in the front?
Oh.
Yeah. That's like a bladder. That's a backup tank that keeps pressure at the pressure these things like.
Huh.
And then our pressure goes into that.
So now it never drops.
Okay.
It's like an aftermarket mod on your BMW.
That's right. Yeah.
We are now resolving what is essentially an annoying failure in the device by something we're incorporating into the rack cart. So what we want it to feel like is this is the most automation-friendly version that can be made of this device.
Okay.
Right? And we even have a newer version where we control the humidity too.
Yeah.
Same devices, but see those 3 little things sticking down?
Sure.
That's because humidity also matters.
Wow.
So why don't we control that too and reduce the failures? So the other thing I want people to experience is these devices working even better than they would at the bench because we've incorporated them into our systems where we can add this stuff.
Right.
Does that make sense?
It does.
And then eventually you'll just remake them as it makes sense. Or the other vendors will, I hope.
Yeah.
Right?
Well,
Like that would be ideal.
to that point, one of my favorite classes I took was around innovation. They talk about looking at the needs of extreme users
Yes.
as a way to figure out where to go with your product. And so I imagine as they hear that, they're like, okay, well, we got to make the pressure better, right? So it's
Think about it ahead of time. Yeah. We are the extreme users. Yeah, like we have certain devices here. I won't name which one, but where we were using it like 70% utilization rate.
Yeah.
And the tech was having to come in, fix it. And they finally come in, they're like, you're outside our operating specs. And we're like, well, I'm sorry. I bought this. I didn't know that you only expect it. It's because like in a benchtop environment, all these lab devices are used like less than 25% of the time.
Wow. Yeah.
Because think about it, right? You're only working 5 days a week.
Yeah.
Maybe only in the lab 5 hours a day.
Yeah.
All right. This thing is running 24/7.
Sure.
Right. I can access it in the middle of the night and hit that thing. So if it's an expensive device, I want to be using it all the time.
Yeah. Fascinating.
Yeah. It's fast. It really is. It's just different. Yeah. Watch yourself on this.
All right.
Yeah.
Yeah.
Gonna come behind you, sorry, bye. Okay. All right, so that's sort of that's the whirlwind tour of the world's largest autonomous lab. And like I said, we'll be at 100 racks by May.
Okay.
So about twice the size of the current system.
Fascinating.
Yeah.
Well, thank you so much for just walking us through that and again showing us where sort of maybe hype and headlines actually meets reality.
Yeah.
With that, would love to maybe just ask you a couple more questions and wrap up our chat today. Does that work for you?
Yeah, works great.
Yeah.
Let's do it.
Okay.
Yeah.
Cool. Awesome. Well, Jason, thank you for that awesome tour. I really enjoy just seeing this come to life, and also,
Or it helps us see it, right?
it does, yeah. And I'm also just dreaming of like what the version 2 of this lab looks like, where you have devices that are kind of built from the ground up for autonomy. I think it's going to be really cool.
Yeah.
So I'm, an MBA. So putting my business hat on, that lab is amazing, but it also, that equipment doesn't come cheap.
Yep.
And so you, as the CEO of Ginkgo Bioworks, so you have been CEO for, it sounds like 18 years or so, almost coming up.
2008.
Yeah.
2009.
You started as, I think, a YC company.
We were the first YC biotech.
Ever biotech.
Yeah, in 2014. In other words, we were a YC company, but we were the first biotech to have ever done the program. It had only been software before. And it was when Sam took over.
Okay.
Mr. Famous, Sam Altman, had took over as president of YC, and he wanted to move the YC model into deep tech. So also in our batch was like Helion Energy, like the fusion company in Seattle. Yeah, well, there were like 10 people at the time. Like it was like really fun. It was a cool batch, but it was like deep tech. Okay. And so, yeah, we had a great experience doing it.
So, deep tech is having a moment again today, and, there may be some, aspiring deep tech founders or current early stage deep tech founders who are listening to this episode. As you reflect on your, long journey with, I imagine, a lot of lessons learned, like what advice would you have for folks who are looking to enter this space and, again, work in the world of atoms as you have?
Yeah, I do talk to lots of deep tech entrepreneurs, right? So Ginkgo's raised more than $2 billion over the years. Like we're from the standpoint of being able to access generalist capital as a deep tech company. I think we have some lessons. And then building a deep tech business. We can talk about that next, right? So just on the like entrepreneur generalist, where it's not bio specific, but just about building hard things. I think one piece of advice is it's not like you're going to ramp into revenues in many of these companies. It's hard to find a business model that does that in the near term, like a SaaS company would have, or like the AI models are doing today. Okay. So that doesn't mean it's the end of the world. Like SpaceX and Tesla did not ramp quickly into revenues. Okay. Right. Like it doesn't have to. Right. So it's just a different playbook. All right. But you need to convince people that you're making progress. So I think one of the key things as a deep tech entrepreneur is what are those milestones along the way that have meaning to the investor? So like a good example in YC, there's a company called Boom Aerospace. It's like trying to make the next Concorde supersonic flight. During YC, they did a deal with Richard Branson. That was like, when this thing is ready, and this is like, they had like a, diagram or something at the time, right? Virgin would love to buy some. That got their seed round done, right? Because that showed that this was like a founder who wasn't sort of the traditional boring, slow aerospace model, but more like the hustle, fast Silicon Valley mentality. And some people will just bet on that because now, and they saw it. They saw it in how they were able to do that deal. That's material, right? Like that was the most important thing I would say that they were able to do during the 12 weeks of YC. They already had the model, the plane, everything else, but like, how do you speak to the generalists? You have to show these steps along the way. So I would say the first thing is, even if there's a certain set of steps that are traditional in the aerospace industry or the pharmaceutical industry or whatever deep tech thing you're doing, can you find some other orthogonal things that help show your chops as a as a Palmer Luckey, or as as an entrepreneur that is really going to upend, as an Elon, as an entrepreneur that will upend these staid industries. Does that make sense? And anything, and be creative. So that's sort of point number one. Point number two, I would say deep tech is more spread out. Like it's not like software lives in California. I mean, a lot of the, look at SpaceX, that all was in LA area, right? Like aerospace is a lot in LA. Robotics is a lot in Boston, actually. Biotech is very much in Boston. And there's other pockets around the country where there's like specialization. Agriculture is all in the Midwest. If you're going to do an ag tech, you should be in Missouri, right? You should be around where like the old Monsanto campus was. Like there's just going to be centers of deep tech excellence that aren't Silicon Valley. And the biggest mistake I see those deep tech entrepreneurs make is they think, I raise venture capital where I am. And the analogy I would give you is like, think about when you're going to take your company public someday, entrepreneur, and you're going to do a roadshow before the IPO. Do you know what city you're going to do that roadshow in? And the answer is, you already know the city. The city is New York City. That's where you're going to do the roadshow. And the reason is not that your company is in New York City. That's where the money is, right? Like that is where the public market investors, the crossover investors that invest in that IPO round and after, that's where they are. And you need to meet them in person. You need to go there and you do a roadshow, right? And you might also, if you're from Boston, do a couple in Boston or Indiana, wherever the hell you are, but you're going to do your roadshow in New York. If you are a deep tech entrepreneur and you are raising a seed round of money in private venture capital, you need to do your roadshow where the money is. And the money is in California. It is in Silicon Valley and San Francisco. You need to go there on an airplane, meet people for a couple weeks, and then everything will be Zoom after that from their house in Tahoe or wherever the hell they are, right? And that is where you should raise your money. That is the biggest mistake I see people make. They do the fundraising on hard mode because the deep tech ecosystem, I see Boston founders make this mistake all the time. We don't have good venture capital here. Like there are some venture capitalists, but it's nothing compared to the volume. Just like there are some public investors here too, but the volume in New York is bigger. The volume in California is bigger by a factor of 100. Why are you making your fundraising 100 times harder? And so those are my two big things. Be creative on the near and milestones and raise money in California. You don't have to be in California to raise money. Like money travels through the internet. Okay. I'm just letting people know. These are the two mistakes I see deep tech entrepreneurs make over and over and over again. And I'm like, guys, why are you doing it on hard mode?
What areas of, let's maybe zoom more into the world of Ginkgo and biotech and so forth, are, if you are right now graduating from MIT, and you had, a question ahead of you of where do I place my efforts? What either kind of areas do you think are especially interesting or exciting, and or maybe areas that would be really good complements to, I think, to the vision that you have of autonomous science? So it's maybe not what Ginkgo is doing, but that would be complementary and really help the ecosystem move forward.
Yeah, okay, it's a fun conversation. So yeah, first off, I would say people should go where they're excited, right? Because I think one other thing is everyone thinks this stuff will happen fast and everything takes forever. So like the biggest advantage Ginkgo has had is staying power among the founders and even some of the early employees too are still here, right? Like it's this sense that we have a thing we're trying to accomplish and we're going to just stick with it for a long enough time. And I think that's obviously true of places like a SpaceX or a Tesla and so on. OpenAI, I mean, yeah, OpenAI has been great now, but like look at the first 5 years of OpenAI, right? Like Sam was running like a nonprofit research institute, right? Like and so I think that you really only can stick it out if you care about it. And yeah, you can do small things, and you can make money, by the way, doing short-term stuff, but you don't get to make like a Google or a SpaceX or an OpenAI short-term. It literally has never happened. So if you're interested in that sort of thing, you pick something that you want to do for 20 years. I think that's actually like the most important thing. Then, okay, now we can talk science and autonomous labs and all that stuff. I think it's super fast. I'd love to, yeah, let's game business models, right? So, I mean, I like to talk about what I'm doing at Ginkgo, but if I look at like the macro question, so for like other entrepreneurs that want to start companies, I think the door's open for a combination of, they don't even have to be geniuses. Like you just need models that are I think basically already as smart as the coding models that can do more or less experimental design, data interpretation, inclusive of data science tools and programming, and draw logical conclusions, and then design more experiments. Those models already exist. Scientists can do that too, but I can generate 30,000 models. Okay, it's very expensive to hire 30,000 scientists, but I could have 30,000 models pursuing 30,000 hypotheses to attack a problem. Next, they need to be able to do lab work. They cannot think it up. That's the thing. That only works in math, right? So now they need to have access. You need autonomous labs that can work with 30,000 models. That's what we're working on, but I think it's going to get there. I don't see a blocker other than engineering work from delivering that. So that will be delivered, and we have enough money, so I think we will deliver it. Okay, so $400 million in the bank, right? So okay, so that's sort of that ecosystem I think will exist. And I think I'll make one last point on this model thing. The way we do science now is basically you run your lab. We're working on the same area. We have two different hypotheses, two different professors. We might do a year or two of work and then publish a result. And then you get to see what I was doing. And we kind of talk at conferences, but it's a little circumspect. These 30,000 agents could be basically passing not just like what they learned, like their conclusions that day, but actually their data.
Including their failures.
100%, including their failures, which literally never get passed in the traditional scientific environment. So there's massive advantages, I think, to doing science this way. So it's like that exists, the robotic lab exists, and let's just imagine you put that in a pot and stew it, and Genesis Mission funds it on the US side, so we all get to try this. The NIH funds it, I don't know, right? Like some companies try it, and we all realize that the rate of scientific discovery with this system is 100 times faster than it was before. Okay, I'm not sure that's going to happen. Let's just say it did. What companies do you start? Like, how do you, like, what are the opportunities in a world where not programming goes 100 times faster, but rather scientific discovery goes 100 times faster? And I then think we have a great intuition about that. Pharma is the closest thing of all the industries of saying, I'm going to go after something that I really don't know if it's scientifically feasible to do it. Like we're going to work on Alzheimer's. Like we might be 200 years from unlocking Alzheimer's, but a pharma company will spend $3 billion on a series of hypotheses to try to unlock that. That is exceedingly rare across industry. It's even just a culture to tolerate that. Right? Like I tried this drug and we spent $500 million and it failed in the phase 3 trial. It turned out to be a bust. No one calls that like leader of that company like a fraud, for example. Right? Like that, even that is a lot of tolerance. Like, are you kidding me? You spent $500 million and it's worth zero? How in any other industry, that feels like something insane must have happened. But we've created both the cultural like architecture and the way to spend, to smartly try things and milestones and all this stuff in therapeutics in particular to essentially try to reliably mine something that's very unpredictable, which is a scientific discovery. So should we be taking that model and roughly applying it to material science and energy, blah, 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 like go down the list of these other places? There are ways, like I don't know, right? Like, but like I think there's an interesting question for like investors, entrepreneurs. If you assume that world is coming, what does it look like? Like, what are the new types of product companies? Like, how does it work? Okay, I want to sell the picks and shovels, but like, I think if you, and so does an OpenAI on the frontier side, I want to sell on the robot side, but then there's a user of that stuff, and I don't know. Like, I think it's interesting. I think it's like it's a really interesting question, and potentially like a ludicrously lucrative one. Because if you're the first one out of the gate with a true breakthrough, electricity, you get to be General Electric. Right? Like these are crazy. These are like the behemoths. You reuse off rockets, you're SpaceX, right? Like there's just, it's really interesting. But I think it's a super exciting time to be an entrepreneur who lives at the scientific edge there, like these deep tech folks who really know what would be a holy grail breakthrough in their field. And if they say, well, that one I kind of feel like if we could put 100 times the effort at it, we'll hit it. In which case I'm going to bet on that happening. And then I'll be, right? Like I'll be Edison, like I'll get there first, right? Like I mean, that's super interesting to me. I don't know, right? But anyway, that I think it's a cool question. I can talk now about how we'll sell picks and shovels, but like that's, I think the more macro, all deep tech entrepreneurs might start to expect that to come and watch it.
Yeah.
But as it starts to feel like it's happening, jump in the pool.
That's great. So, on your own personal journey, so one of the highlights as we started off in the beginning, it was going public and being a publicly traded company. That comes with its own new set of responsibilities and challenges, activist investors, lawsuits, but also opportunities to like partner at a larger scale and hire great talent and so forth. Like what have you learned from that experience that you might pass on to folks who maybe aspire to be in your position?
Yeah, I'll talk about that, and we should circle back on the business model side too. So I think my take on being public is I like it from the standpoint of you have more access to capital, right? So we're able to raise a ton of money at the listing. That's very valuable for a company like us that's making a lot of investments in technology. So that's like sort of like number one, right? Like, and this has changed a little bit because the private rounds have become so massive, right? But even then, that's a little bit rarefied air, right? Like you can raise it if you're Sam, you can raise it if it's like these companies that are crazy, or Elon, whatever he's doing. But then for a lot of entrepreneurs, if you really want to tap like really huge pools of capital, the better way to do it is to go public. Number two, I like the idea of ultimately growing this company, which I think is going to, I want to build a Google, right? Like I want to build a $1 trillion company. There's a lot of growth from here to there. There's a lot of growth even from where we went public, which we're down from there to being that company. So like, I want people to be able to come along for that. So one of the things I like about being public is like anyone can invest, right? So I like that. Like, I think that's cool. I think you do have to be careful with how you structure the company to make it like a little more difficult to get pulled around by your nose, right? And so you can do that with super voting shares, things like that help with that. But like so I think you want to think carefully at the point of going public, how you're going to like basically like set your charter and stuff. And then I think folks like a Buffett, like the sort of like Amazon folks, like there's like a view on how to think about what you as a leader of a public company should be doing. And the short form of it is building a great business. It is not like talking to analysts, right? Like talking to analysts does not make you a better company, right? And so you have to be very judicious about that, and not, I think a lot of people go public and they start to think their job is to talk to investors as the leader of the company, and it is absolutely not. It is to build a great business. Does that make sense? So those are like, there are pitfalls to avoid, but I think if you can avoid them and set things up properly, it's just categorically better. And then we've done things over the years, like the robotics technology we acquired originally 4 or 5 years ago now from a company called Zymergen. We were able to do that basically because we'd gone public and had, shares to use and dollars to use. And so like, that's the kind of thing I couldn't have done if I was still private. Does that make sense? And that's been now hugely important given the direction the company has taken and the world has taken.
So as a company that's gone public in this space, that's at the forefront of like autonomous science, I'm curious, like, how do you think that, as an OG in this space, at this point, I might say, like, Ginkgo is perceived in the industry?
So I think we're like an unknown a little bit when it comes to thinking of us as like automation, right? So what people know us as is like kind of fancy genetic engineering of cells. We have over the years done 250 research partnerships, Bayer, Novo Nordisk, Pfizer, all down the list, right? Starting in things like industrial biotech and then agriculture and then pharmaceuticals. And that was sort of like our business model for 10 years. We now call this our solutions business. We still do these deals, like Bayer's still a big customer, Novo's still a big customer. We add new solutions deals pretty regularly. But the like center of gravity of the business in the last 2.5 years has moved to tools, which means a little more like a Thermo Fisher. And so people don't think of us as a Thermo Fisher. They're like, I didn't know I could buy your technology, Ginkgo. Like, I thought that was like super secret in-house stuff that you would then try to do a deal with me to get a royalty on some project. And I'm like, no, if you want it, I'll install it, right? So there's a little bit of a rebranding of ourselves as a tools company. And that's actually, I think, the biggest misunderstanding from like the standpoint of like customer side of the house, if that makes sense. But we're getting the word out. Like this SLAS was awesome. We had 550 people come through and take the tour. I just got to take you on. Yeah, it was like a madhouse. Yeah, and so I think that is changing real fast. But that's been I'd say misunderstanding about us. Like the easy way to think about it is if you see some technology at Ginkgo you really like, you can buy it, right? And you can use it in your own lab and use it to accelerate your own scientists. That's what's new in the last couple of years at Ginkgo versus the first 15.
You said you wanted to touch on business models. Yeah.
Yeah, because you brought up like Emerald and like the cloud lab thing and just sort of like what's worked and not, right? So, okay, so we are, for the first long time in the company, we worked on a business model. It's roughly a company like Adimab in the biopharma industry where they make antibodies for people. So it's like you call them up, you pay them X dollars of research fees, it's not a big number, and then they develop you an antibody, you take it, you try to go to market with it. Yep. If you're successful, you'll give them an extra milestone payment at phase 2 or phase 3, and then you'll give them a royalty for the life of the drug. And so they're kind of like outsourced research with a tail. And we've kind of ran that business model, but for like any genetic engineering, it just takes a long time. That's the basic problem with that business model. It's otherwise pretty interesting. The business model we didn't want to do was become a drug company. So we didn't want to vertically integrate into the end product. We wanted to try to find a horizontal tech platformy kind of business model. Because my feeling was once you become a drug company, you better get ready to go, right? Like that's a competitive industry and it can't be about figuring out how to sell this technology to that guy or something like that. It's about what's the next drug to go after and how do you defend your turf and all that stuff.
Pretty binary outcome.
It's a real, and it's just it's a real game that really orients around the drug and the patients that go to market, the whole thing. And it's just not really about the technology that was 7 years ago made you that drug. It very quickly moves elsewhere. And so I was like, that does not support what we're trying to accomplish, which is to make biology easier to engineer over a decade of technological work to like basically get like a Moore's law going. Yeah. And so I was like, we need a different business model. And so first I tried the royalty thing and the main thing was the timing. And so now I'm trying the tools thing. So it's like, I will like install the robots, or if you don't want to buy a system from me, I will run my system like a cloud. And so you can get CRO services from me where I'm not taking a royalty or anything. And we just launched cloud.ginkgo.bio on Monday, where you can now go in and for like $39, you can do a protein expression experiment on that setup and the plates will run and you'll get data back.
You heard it here first.
Yeah, it's really neat. And so that is a thing that did get tried by places like Emerald Cloud Lab, which is still in business, by the way, and Transcriptic Stratios, which is not. And it's been a historically difficult business model to do. And so the trick is the basic problem is, does the scientist trust you to do the experiment? And if the experiment doesn't work, did it not work because you did the experiment wrong or because they designed a bad experiment? And at the bench, they wouldn't think twice about it. They'd be like, oh, I'll do it again. Because they did it themselves. So it's like, well, I guess it was my fault, right? And so I'll just try again. And then if they do it twice and it fails, they'll be like, oh, my experimental design is wrong. I got to change my experimental design. But you can get into this like friction with the service provider where the expectation is really a successful experiment outcome, not a successful experiment execution. Does that make sense, the difference? And so we're sort of like warming up to that by putting on Ginkgo certified protocols that we already know work great. And so we're going to you're definitely going to get the result that you want. And then we're going to, over time, as we build trust with people, allow variation. Oh, you want to make a slight change on that? Go ahead, right? And then eventually, you're just going to log in like one of my scientists does and order whatever experiment you want. But that'll be a journey. Does that make sense? But those are the two halves of the business. We're basically either build you one or run ours. And yeah, we'll see. But all of that is us basically being able to prove out that autonomous labs can do that variable work, and I can sell it to you in 2 ways.
Well, what I'm hearing in there, which is what I hear from, I think, folks who are business-minded, is we're experimenting ourselves and trying to figure out where we can get product market fit with this idea to support this, I think, aspirational vision to benefit humanity. A book I read a few months ago was The $1 Billion Molecule.
Oh, yeah,
about the rise of
Biotech.
Gave me, as a non-scientist, an appreciation for how much storytelling is involved and how much kind of improvisation is required to get one of these companies off the ground.
Yeah.
But then obviously when that's successful, we all benefit from it.
Yeah, that is right. Yeah. It's a great industry.
With that, Jason, just really appreciate you sitting down with us today and giving us this tour. It's really special for me, and I know for the audience here. As somebody who I think still has that sci-fi, type kid in me as well, also from the same generation.
Yeah.
It's really cool to see somebody actually trying to make that a reality for us. So thank you for that.
Yeah. Thank you for spending the time with us and coming to see it. I think it makes a big difference. So absolutely, man.
Okay, take care.
Appreciate it. Cheers.
Bye.